Okay. Glycolysis has resulted in the formation of what it has resulted in the formation of pyruvic acid molecule. So 3 carbon compound CS3 CO COOH C3H4O3. Okay, this is pyruvic acid. 2 molecules we have got. So the fate of this what we call pyruvic acid is determined either there is presence of oxygen or there is absence of oxygen. Okay, say glucose to pyruvic acid. This is 6 carbon. This is 3 carbon, 2 molecules. Okay, this process what we call it glycolysis. It occurs in cytoplasm of the cell. Now, this pyruvic acid, in presence of oxygen, it undergoes complete oxidation. It undergoes complete oxidation. So when it undergoes complete oxidation, we are going to get carbon dioxide and water with the release of huge amount of energy. So this part is, we can refer it as aerobic respiration. This part, the fate of the pyruvic acid further, it is determined either by the presence of oxygen or the absence of oxygen. If oxygen is there, it undergoes complete oxidation and result in the formation of carbon dioxide and water with the release of huge amount of energy. Okay. So in case of aerobic respiration, this pyruvic acid will undergo 2 steps. One that is Krebs cycle, and another one that is electron transport system and oxidative phosphorylation. These are 2 steps it has to undergo this pyruvic acid. Okay. Suppose in absence of oxygen, it can undergo what you call fermentation. When it undergoes fermentation in absence of oxygen, it can form 2 types of products depending on the kind of the fermentation. So one is this is ethyl alcohol, and there is release of less energy. So here is more energy, that is 36 ATP molecules. So usually 6 carbon dioxide, 6 water are formed. More energy is released. So here less energy, that is only 2 ATP molecules are produced. So ethyl alcohol is formed. So this is one product. If it is alcoholic fermentation, then ethyl alcohol is produced. Okay, suppose if it is lactic acid fermentation, then what will be produced? C3H6O3. This is lactic acid. No carbon dioxide liberation, and as usual it release only 2 ATP molecules. So here 686 kilocalories of energy, and here only around 56 kilocalories of energy is available. So we have to discuss now this fermentation. After fermentation, we will move on to this mechanism of aerobic respiration, that is Krebs cycle and electron transport system. All that. So anaerobic respiration, incomplete breakdown of glucose either into ethyl alcohol or lactic acid. Anaerobic breakdown. Okay. In absence of oxygen, the respiratory substrate is incompletely broken. Okay. Either into ethyl alcohol or lactic acid with the release of less amount of energy. So anaerobic respiration occurs inside the cell. with the help of intracellular enzymes. Respiratory substrate is present inside the cell. Enzymes are present inside the cell. So where it is incompletely broken because of absence of oxygen. Now, we will get fermentation. Fermentation is a kind of anaerobic respiration. It is a kind of anaerobic respiration which occur outside the cell with the help of extracellular enzymes. Fermentation is carried out in a specially designed tank or fermentation vessel, or even we call it as bioreactor. So fermentation is a type of anaerobic respiration only. Anaerobic respiration. Okay. So substrate is incompletely oxidized into either you will get the ethyl alcohol, or we will get what we will get lactic acid. Okay, so it is a type of anaerobic respiration where substrate is incompletely broken or oxidized either into ethyl alcohol or lactic acid. This process occurs usually outside the cell. Means it is the extracellular process. It is the extracellular process outside the cell with the help of extracellular enzymes. Extracellular enzymes. Usually it occurs outside the cell with the help of extracellular enzymes. So based on the nature of the product formed here, we have mainly two types of fermentation. One is alcoholic fermentation, and second one is lactic acid fermentation. Alcoholic fermentation is the basis of beverage industry. Beverage industry is the alcoholic beverages are produced. Beer, wine, brandy, whiskey, rum, vodka, gin, different types of alcoholic products are produced. Okay. So particularly preparation of beer, preparation of wine, preparation of champagne, that can involve mainly alcoholic fermentation. Okay. These alcoholic drinks, they have less percentage of alcohol. Especially beer, wine, champagne, red wine, all these what we say. Other products, they have very high percentage of alcohol. They are produced through distillation process. Now, lactic acid fermentation is the basis of dairy industry. We have dairy farm or dairy industry where cattle rearing and breeding is carried out. We produce a different kind of milk products. So, lactic acid fermentation is the basis of dairy farm or dairy industry. So, first, we will discuss alcoholic fermentation. So, in this fermentation, ethyl alcohol is the major product. Right? So, alcoholic fermentation, it involves three steps. First time this alcoholic fermentation was first observed by Louis Patch. Louis Patch, first time he discovered or observed this alcoholic fermentation. This alcoholic fermentation is carried out in a fermentation chamber with controlled conditions. Now, this alcoholic fermentation make use of a very important what we call microbe. That microbe is yeast, a unicellular fungus. A unicellular fungus. This fungus is the source of enzymes like invertase. This invertase breaks sucrose into glucose and fructose. It is a source of yeast is a source of enzyme like invertase, which breaks sucrose into glucose and fructose. Then another important enzyme is there in the yeast that is called zymase enzyme complex. Zymase enzyme complex that is mainly important for this alcoholic fermentation. Remember. Now, three steps of alcoholic fermentation. One is glycolysis. Second one, decarboxylation. Third one is reduction. So these are the three steps in alcoholic fermentation. See, first step, annual glycolysis. Okay, so in glycolysis, what happens? Glucose. Okay, glucose is partially broken down into two molecules of pyruvic acid. Right? So this glucose is broken down into two molecules of pyruvic acid. Two molecules of pyruvic acid, 3 carbon compound. This is the 6 carbon compound, C6H12O6. Okay, it splits into two molecules of CS3CO, COOH, right? And there will be a collection of 2 ATP and 2 NADH. This is the simple overall reaction of this glycolysis. This is glycolysis, partial breakdown of glucose, 6 carbon sugar into two molecules of 3 carbon pyruvic acid. Okay, it is written as C4 C3 H4O3. Okay, so we got pyruvic acid, ATP, and NADH. This is the first step, that is glycolysis. Now, the second step, that is what is it? Decarboxylation. See, this glycolysis is not a single step. We have written single equation, but this step occurs in 10 steps in linear sequence fashion. These 10 steps occur, and that result in the formation of the pyruvic acid. Now, decarboxylation. So, second step, that is decarboxylation. This pyruvic acid is there. Two molecules of pyruvic acid, carbon, two molecules. Okay, this pyruvic acid will undergo decarboxylation. It undergoes decarboxylation with the help of pyruvic decarboxylase enzyme. Pyruvic decarboxylase enzyme. Okay, then it results in the formation of two molecules of acetaldehyde. We get two molecules of acetaldehyde we will get, aldehyde we will get. And this is the process by which the carbon dioxide is liberated. It's the process by which carbon dioxide is liberated. So alcoholic fermentation, overall reaction, what you will write here? C6 H12O6. Okay, you will write two molecules of C2H5ONZ plus 2 carbon dioxide plus 2 ATP molecule will get. So this carbon dioxide is coming from decarboxylation step. The carbon dioxide which is liberated now is coming from decarboxylation step. And ATP which are produced, they are produced in glycolysis itself. They are produced in glycolysis. Okay. So what we got now? Two molecules of acetaldehyde we got at the end of decarboxylation. So it is catalyzed by pyruvic decarboxylation. Okay. Now, third step. What is the third step? What is the third step? Reduction. So in the reduction step, what happens? This acetaldehyde which is formed here, two molecules of acetaldehyde will be reduced by the reduced coenzyme which is produced in glycolysis. NADH, nicotinamide adenine dinucleotide reduced form. It is a reduced coenzyme. So this reduced coenzyme will reduce this acetaldehyde into two molecules of ethyl alcohol. This is catalyzed by alcohol dehydrogenase enzyme. Alcohol dehydrogenase enzyme. So this is ethyl alcohol. This is ethyl alcohol. So this is now through three steps there will be synthesis of ethyl alcohol in alcoholic fermentation. So this is catalyzed by here is not right. Zymase enzyme complex. So this is it. Glucose, ethyl alcohol. This is overall reaction of alcoholic fermentation. Overall reaction. Okay. So incomplete breakdown of glucose in the absence of oxygen. In the absence of oxygen, with the help of zymase enzyme complex. Zymase enzyme complex where it is present in yeast. So use of yeast is inevitable here. Right? To produce ethyl alcohol, carbon dioxide is released, and two ATP molecules. So the number of ATP molecules produced will be less. As I told in the previous class, this glycolysis or fermentation, it can account only for 5% of the ATP produced. Glycolysis, I told glycolysis account for only 5% of the ATP produced out of the total ATP. And I told it accounts for less than 7% of the energy released. Whatever the energy which is stored in glucose. Complete energy will not be released. Less than 7% of the energy stored which is there in the glucose is released from fermentation process or from glycolysis. So energy release is comparatively less. So it is a less efficient process with respect to the amount of energy released during this fermentation process. So that we remember, right? So this alcoholic fermentation is commercially used in the preparation of the alcoholic beverages. Now listen, this fermentation continues. This fermentation continues by the action of yeast with the help of its enzyme. Okay, until 10 to 13% of alcohol is formed. Alcoholic fermentation can continue in the fermentation chamber or fermentation vessel up to 10 to 13% of ethyl alcohol is produced. Afterwards, higher percentage of this ethyl alcohol can be toxic to yeast, can be toxic to yeast. As a result, enzymes will get inactivated. So fermentation can stop. Alcoholic fermentation continues until 10 to 13% of ethyl alcohol is produced during fermentation process. Rise in alcohol can be toxic to the yeast. Enzymes get inactivated, hence fermentation stops. Now, as I mentioned earlier, we have other alcoholic beverages. Okay, so this beer, wine, cider, these are all undistilled alcoholic beverages. They are undistilled alcoholic beverages. Whereas brandy, whiskey, rum, vodka, gin, etc. These are all distilled alcoholic beverages. In them, see, they have around 8 to 10% of alcohol. In them, alcohol percentage is variable. It can be variable between 40 to 55%. These 40 to 55%, that brandy, whiskey, rum, vodka, gin, they have high percentage of alcohol. So to produce such alcoholic beverages, fermentation will not work out here because I told fermentation can continue up to this much percentage of alcohol produced in the fermentation vessel by the action of yeast with the help of its enzyme. Later on, this higher percent of alcohol can become toxic to the yeast. Yeast enzymes get inactivated, fermentation starts. Then how can we produce such what we call alcoholic beverages? These are produced through distillation process. Remember, these are produced mainly through distillation. So distillation process can increase the percentage of alcohol in these beverages. So this is all about alcoholic fermentation. Now moving on to the next one now, that is lactic acid fermentation. Lactic acid fermentation. So this lactic acid fermentation, I told it is the basis of the dairy farm or dairy industry. Okay. So here, usually in alcoholic fermentation, glucose is the substrate here. That glucose can be derived from sucrose. Sucrose, with the help of inverted enzyme, is broken into glucose and fructose. So then it acts as a substrate. Here in lactic acid fermentation, lactose sugar, lactose sugar can be substrate. Lactose sugar can be the substrate. So usually what happens is we will need to O11 hydrolysis. That's the lactose. Okay. This lactose sugar on hydrolysis, it can yield what it can yield C6H12O6. This is glucose. And one more product you will get that is galactose. Galactose. This also same formula. Slight difference will be there in its structure. Glucose and galactose you will get. So this can act as a substrate for lactic acid fermentation. Lactic acid fermentation is carried out by the group of bacteria. What is that? LAB, lactic acid bacteria. Remember, This is lactic acid bacteria. See, fermentation is carried out by certain bacteria, okay, certain fungi, and you can see in what we call the muscle cells. Muscle cells, skeletal muscle cells in animals and in human beings, you can see this kind of fermentation, lactic acid fermentation. Unicellular eukaryote yeast. Unicellular eukaryote, it is fungus. Okay, so certain bacteria, fungi, muscle cells, skeletal muscle cells of the animal or human being, especially these muscle cells which are involved in heavy exercise, heavy running. Okay, there you can see this lactic acid fermentation. So basically, fermentation is carried out with the help or present in microbes, carried out with the help of these microbes. So lactic acid bacteria play a very important role in lactic acid fermentation. So we have different lactic acid bacteria. One is Lactobacillus lactis, Lactobacillus lactis. Okay, then Lactobacillus acidophilus, acidophilus. Then you have Streptococcus lactis. These are LABs, lactic acid bacteria, that remember. Okay, so the glucose which is formed here, overall reaction of lactic acid fermentation, C6H12O6, it yields two molecules of C3H6O3 plus two molecules of ATP. There is no liberation of carbon dioxide in lactic acid fermentation, remember. Glucose. So here, this is lactic acid, C3H6O3. It is also written as CS3, CHOH, COOH, 1, 2, 3, C3, 3, 4, 5, 6, 6, 4, 3, 1, 2, 3. This is the formula of lactic acid, remember. CS3, CHOH, COH. This is the overall reaction of lactic acid fermentation. So it is carried out with the help of lactic acid bacteria, LABs. So in the case of lactic acid fermentation, there are only two steps. In alcoholic fermentation, three steps are there: glycolysis, decarboxylation, and reduction. There are only two steps. In lactic acid fermentation, only two steps are there. Okay, one and it is glycolysis, and the second one here directly reduction. The second one is directly reduction. So first we will write a glycolysis reaction. So in glycolysis, what happens? Glucose is partially broken down. Glucose is partially broken down. Okay. So this is the glycolysis. What you will get here? CS3, CO, COOH, two molecules, three carbon compound. What is this? Pyruvic acid plus what will you get there? Two molecules of ATP. and plus what you will get? Two molecules of NADH2 will get. Two molecules of NADH2 we are going to get. Okay, so this is the glycolysis which occur in 10 steps, right? So we got pyruvic acid, 285, 2 NADH2. So this is the reduced coenzyme. Reduced coenzyme, nicotinamide adenine dinucleotide. Now directly what happens in the second step? So this is the first step. In the second step directly, this pyruvic acid will be reduced. Pyruvic acid will be reduced with the help of this reduced coenzyme. There is no decarboxylation step, and there is no decarboxylation step in lactic acid fermentation. There is no liberation of carbon dioxide. So now you write that is second step, that is reduction. Reduction. So two molecules of pyruvic acid. Okay, this pyruvic acid, how it is written? C3H4 O3. This also we can write. This will be reduced by whom? Reduced coenzyme. This reaction is catalyzed by, you have to remember this enzyme, very important enzyme, lactate dehydrogenase enzyme. These enzymes you remember in alcoholic fermentation, decarboxylation step, pyruvic decarboxylase enzyme, then reduction step, alcohol dehydrogenase enzyme. So here it is lactate dehydrogenase enzyme. So we are going to get two molecules of CH3, CHOH, COOH. CH3, CHOH, COOH. Okay. Plus what will get? What will get? 2 NAD plus. 2 NAD plus will get. So 1 minute. This is lactic acid. This is lactic acid, or what is the another formula? C3H6O3. Two molecules of lactic acid are produced here. So this is how this anoxidation. This anoxidation releases two hydrogens. These hydrogens are added to it. It will get CH3, CHOH, COH. So this will become lactic acid. So this is how lactic acid fermentation is carried out with the help of what we call LAB, lactic acid bacteria. Right? So especially in case of animals and in case of human beings, whenever they are involved in heavy exercise, that the muscle cell will not get adequate oxygen. The muscle cells, skeletal muscle cells of the legs do not get oxygen properly. So that the muscle cell, when it do not get oxygen properly, so in absence of oxygen, muscle cell will perform what it will perform? Lactic acid fermentation. So in the muscle cell, when lactic acid fermentation occurs, that lactic acid which is there, this lactic acid dissociate into lactate ion and proton. Lactate ion and proton in the muscle cells. Protons concentrate in the muscle cell. Because of concentration of the protons in the muscle cell, the pH of the muscle cell will become acidic. Okay, it will become acidic. So this leads to muscle cramp, common in sports. Muscle cramp, very painful it is. Muscle cramp, very painful because of this lactic acid fermentation it is produced. Remember this. Lactic acid which is produced It dissociates into lactate ion and proton. Protons concentrate in the muscle cell, leading to what we call a decrease in pH. pH will become acidic. This condition results in extremely painful condition that we call it a muscle cramp. Very common in athletes, in tennis player, in cricket player, football player, etc. Sports people, very common. So whenever the muscle cramp is there, we have to give the rest to that particular part. Okay, then automatically within 1 day this cramp can be cured. When it gets enough oxygen supplied to that muscle, then it shifted to aerobic respiration. As a result, this cramp can be treated, cramp will be cured here, right? So this is about lactic acid fermentation. So these two types you have to remember, very important. Overall, fermentation occurs in microbes like bacteria, fungi, unicellular eukaryotes. Okay, even this lactic acid fermentation can occur in higher animals, including human beings. This fermentation releases only less than 7% of the energy stored in glucose. It contributes nearly around 5% of the ATP out of the total ATP production, usually what we see in case of aerobic respiration. Okay, so this is all about fermentation process. Both are commercially important because we have dairy industry, one very useful industry we have. Lot of export we do, milk products, and another one alcoholic beverage. Okay, these are also exported to other country with the help of this alcoholic fermentation carried out in fermentation chamber. All that. The raw material you can use different raw grape wine is made from grape juice, cider is made from apple juice. You can use barley, you can use molasses. Molasses is the dark sugary syrup left after the crystallization of sugar in sugar factory. When molasses undergo fermentation, that is why we get a very foul smell when we pass through sugar factory. Whenever we are passing, you go to this Alicate B sugar factory there, factory will be a little far. But at that time only, what happens is we start getting foul smell, bad smell because of this fermentation of the molasses. So these are important LABs which are involved in curdling of milk. These are involved in curdling of milk. Milk and sugar. Milk sugar. Is it milk sugar? Lactose. Okay. So conversion of milk into curd is carried out by such LABs. The lactic acid bacteria can act as probiotics, live microbial supplement, very good for your gastrointestinal tract, all that. So as there is no decarboxylation step in lactic acid fermentation, there is no release of carbon dioxide. Remember this. So moving on to the next aerobic respiration, very important part. Aerobic respiration. We have already learned the meaning of aerobic respiration. So aerobic respiration, what happens here? Complete oxidation of. So aerobic respiration is complete oxidation of glucose, that is respiratory substrate. Glucose is the commonly used respiratory substrate. So complete oxidation of glucose into carbon dioxide and water. with the release of high amount of energy. Okay, so this complete oxidation of glucose into carbon dioxide and water with the release of high amount of energy occur in presence of oxygen. Occur in presence of oxygen. That's what we are doing. Okay. See, at the end of glycolysis, which occur in the cytoplasm of the cell, we got a pyruvic acid. Okay, C3H4O3. We got there two molecules of 3-carbon compound pyruvic acid. This pyruvic acid is produced in cytoplasm. Another name of cytoplasm is cytosol. Cytosol. Correct? Cytosol. Okay. This pyruvic acid is now transported to. It is transported from cytoplasm. It is transported to into mitochondria. It is transported into mitochondria. In the mitochondria, the aerobic respiration reaction occurs. In the mitochondria, aerobic respiration reaction occurs. So in the mitochondria, you have a part that is matrix. Matrix. It is also referred as stroma. It is not stroma or matrix, what we learned in the chloroplast structure. Okay. So in the matrix, second step of aerobic respiration. First, what is the first step? Glycolysis. Glycolysis do not use any oxygen. It is common step to both aerobic and anaerobic respiration. So in matrix, there is Respiration. This is the second important steps of aerobic respiration. Then now, in the inner membrane of mitochondria, okay, that we call it a cristae, plural, cristae, singular, crystal. You have learned this mitochondria. In cell chapter, like this. Okay, so these you call them as double stranded DNA will be there, semi-autonomous cell organelle, 70 years ribosomes will be there. Okay, this is the double stranded DNA. This is outer membrane, this is inner membrane. Inner membrane is folded into finger-like projections, extending into the matrix. These are referred as crystals. Okay, so this part, which is there, yeah, this part, that is matrix. In the matrix of mitochondria, there is a peptide. And here, on the crystal, okay, if you take a single crystal like this. This is crystal. Okay, so on the crystal, you can say third important step. Okay, that is electron transport system. Terminal oxidation, what we call electron transport system, and oxidative phosphorylation. Electron transport system and oxidative phosphorylation occur where it occurs? Yeah. Okay. So different electron carriers will be there. Yeah. Different complexes will be there which act as the electron transport particles. And one important one is there here. Like this particle. This is called F0. This is the F1 particle. This is involved in oxidative phosphorylation. F0 F1 particles involved in oxidative phosphorylation, and different complexes which are there, they are involved in electron transport system. Here also there will be concentration of protons. Protons will concentrate here. There is verified limit. Protons will concentrate. Chemiosmotic hypothesis we learned chemiosmotic hypothesis is photosynthesis, how ATP are produced. Okay, so here protons will be accumulated in intermembrane space or intracrystal space. So these are the two important steps of what is called aerobic respiration. As a result, what happens? This pyruvic acid is completely decarboxylated. Pyruvic acid here in aerobic respiration. Glycolysis will produce pyruvic acid. Okay, so this pyruvic acid is completely decarboxylated. Completely decarboxylated to produce carbon dioxide and water. So during aerobic respiration, which occur in where it occur the steps of aerobic respiration, mitochondria. Okay, so pyruvic acid is transported to pyruvic acid is transported from cytoplasm to mitochondria. This pyruvic acid first it will undergo preparatory step. Preparatory step or link reaction. We will come to know what is link reaction. Okay, it undergo preparatory step for link reaction. So what happens here? This pyruvic acid, this preparatory step for link reaction occur in the mitochondrial matrix. So pyruvic acid is converted into acetyl coenzyme. Remember this project. How many molecules of pyruvic acid are there? Two molecules. This now keep it in mind. So we have two molecules of pyruvic acid. They undergo preparatory step for link reaction. This is a 2-carbon compound. This is 3-carbon compound. So from two molecules of pyruvic acid, how many molecules of acetyl coenzyme are produced? Two molecules are produced. Two molecules. This is preparatory step for link reaction. Okay. Now, after the preparatory step for link reaction which occur in matrix of mitochondria, then there will be Krebs cycle. This Krebs cycle also occur in matrix of mitochondria. Matrix of mitochondria. So during this, what happens? This acetyl coenzyme is completely oxidized. It is completely oxidized. Okay. So when it is completely oxidized, so it is going to release carbon dioxide molecules. It is going to release carbon dioxide molecule and also it can produce water dioxide. It can produce few number of water molecules. So what happens when oxidation of acetyl coenzyme occurs? When oxidation of acetyl coenzyme in stepwise fashion, it is oxidized. It is oxidized. Okay, when there is an oxidation process, okay, so this Krebs cycle here also 10 steps will be there. Remember, these 10 steps occur in the form of a cycle in mitochondrial matrix. Okay, so there is a liberation of carbon dioxide and water molecule are formed, and there will be formation of what is called reduced NADH2 and FADH2 during this process. During this oxidation, acetyl coenzyme will undergo complete oxidation and turnover. During this, hydrogen atoms are removed. Hydrogen atom is removed. So these hydrogen atoms can be used to reduce oxidized nicotinamide adenine dinucleotide and oxidized flavin adenine dinucleotide. FAD is flavin adenine dinucleotide. Flavin adenine dinucleotide. These are oxidized coenzymes. When there is a proton and electron, they get reduced to NADH2, FADH2. So these are produced during this Krebs cycle. So during this Krebs cycle, okay, what happens? Acyl coenzyme, which is found, it is completely oxidized. When it undergoes oxidation, the hydrogen atoms which are removed, that they will be used to reduce the NAD and FAD. We get reduced coenzymes such as NADH2 and FADH2. So this is the second important step, that is Krebs cycle is occurred in aerobic respiration. So it is followed by the third step. Third step, what I said, electron transport system. So during this electron transport system, what happens is the reduced coenzymes are there. Which are the reduced coenzymes? So during electron transport system, the reduced coenzymes such as NADH2, FADH2, they are oxidized. They are oxidized. So they are oxidized means what they release NAD plus, 2 protons and 2 electrons they can release. And this what it can release FAD plus, it releases 2 protons and 2 electrons. Okay. So these protons and electrons are found. Okay. So the electron which is proton which is released now, these electrons take part in electron transport system. Electron transport system. These protons which are released, these protons can go into that intracrystal space. This, yeah, yeah. Okay. So these protons and the electrons will be transported. Electrons enter into electron transport system. Terminal acceptor. Terminal acceptor of these electrons is oxygen. Oxygen receive proton, oxygen receive electron, and it becomes metabolic water. It becomes metabolic water. When you learn the process, you will come to know. Just to remember, when they undergo oxidation, they release proton and electron. These electrons enter electron transport system. These electrons are finally accepted by oxygen. It receives proton also and get reduced to metabolic water. Okay. So during this electron transport system, using such protons, proton motive force is produced. Proton motive force is produced. So using this proton motive force, what is synthesized? ATP. So ETS, electron transport system, and oxidative phosphorylation. So this oxidative phosphorylation, it synthesizes what it synthesizes? ATP. So electron transport system, there the electrons which are removed from the oxidation of these coenzymes will be transported and finally it is accepted by oxygen to get reduced as water. And there will be synthesis of ATP. The mechanism involved is oxidative phosphorylation. So these events occur mainly in aerobic respiration. So first we will start this. That is preparatory step. Without a preparatory step, Krebs cycle will not commence. Pyruvic acid directly will not enter into Krebs cycle. Pyruvic acid has to be converted into acetyl choline. Gateway step, link reaction, preparatory step, whatever is happening. So this reaction occurs in mitochondrial matrix. So pyruvic acid which are formed in the cytoplasm, they are transported to pyruvic acid are transported to mitochondrial matrix. How many pyruvic acid molecules? Two molecules. How many carbon compound? Three carbon compound. So after entering into mitochondrial matrix, it undergo a process. That step is referred as oxidative decarboxylation. What it is called as an oxidative decarboxylation step. It is called as oxidative decarboxylation step. So in this step, what happens is pyruvic acid, 3 carbon, 2 molecules. Okay, 2 molecules of coenzyme A plus 2 molecules of NAD plus pyruvic dehydrogenase. Very important enzyme, remember, enzyme complex. Here, thymine pyrophosphate, lipoic acid, magnesium ion, thymine pyrophosphate, TPP, thymine pyrophosphate, thymine pyrophosphate. These are all the 5 cofactors which are required. 5 cofactors which are required in preparatory step. Which are those? Thymine pyrophosphate, lipoic acid, magnesium ion, coenzyme A. This is coenzyme A. Okay, and NAD plus oxidized 5 cofactors: NAD, coenzyme A, thymine pyrophosphate, lipoic acid, magnesium ion. 5 cofactors with the help of very important enzyme, that is pyruvic dehydrogenase enzyme complex. Okay, so this preparatory step or link reaction occurs. This pyruvic acid is decarboxylated. So 3 carbon compound to 2 carbon compound will get. So 2 molecules of acetyl coenzyme. This is the 2 carbon compound. 2 molecules you will get plus 2 molecules of NADH2 we will get, and 2 molecules of carbon dioxide are liberated. From 1 molecule of pyruvic acid, 1 carbon dioxide. 2 molecules of pyruvic acid are there, we will get 2 carbon dioxide. From 1 molecule of pyruvic acid, 1 NADH2, 2 are there, so 2 NADH2. So oxidation occurs. When oxidation occurs, that this coenzyme A is converted into acetyl coenzyme A. That is 3 carbon compound is converted into 2 carbon compound. Okay, this is due to decarboxylation step. Then oxidation means where the removal of hydrogen, and those hydrogens are added to NAD plus and it gets reduced to NADH. So NADH2 is produced due to oxidation process. NADH2 is produced due to oxidation, removal of hydrogen that results in addition of hydrogen to this, and it gets reduced to coenzyme. So in the preparatory step, 2 molecules of acetyl coenzyme are produced. 2 carbon dioxide molecules are liberated. Okay, and 2 molecules of NADH2 are produced. So here you try to remember this is preparatory step or link reaction. This acetyl coenzyme is known as connecting link between lipolysis and Krebs cycle. Connecting link between lipolysis and Krebs cycle, S-type coenzyme, which now directly enters into Krebs cycle. Remember this. Okay. So we continue tomorrow.